Alright, so this is my meiosis simulation video, and to start, interphase comes in three main stages, which includes G1, S, and then G2. G1 and G2 are both just stages of growth, so those are not very important to exactly highlight. However, when we look at interphase early, or G1, we can see that there is just chromatin, which is just a mass of genetic material composed of mainly DNA and proteins within the nucleus. It is not coiled up, paired, crossed, or anything, but rather just floating. In meiosis, we start with a diploid, meaning 2n, or having two sets of chromosomes in the cell. In our case, we can see that the diploid number is 16. And now, moving on to S, we can see that the cell is bigger as it grows in G1. However, the main thing to keep in mind is that the DNA has replicated. This replication is important as, without it, we can see that cell chromosome numbers would keep on getting smaller after each division. At this point, the replicated DNA is identical to the one from G1. And skipping G2 because that's really just where the cell grows, we go to prophase 1, and here a couple things happen. We see that the chromatin condenses and then coils up to form something called a chromosome. These chromosomes pair up with their duplicates to form sister chromatids. However, the difference between mitosis and meiosis is that here they also pair up with their homologous partners. A homologous pair of chromosomes can be thought of as chromosomes that have the same length, sequence of genes, and general format of genes. However, they contain different alleles. Specifically, this pairing of homologous chromosomes is known as synapsis. We can see, for example, that all of the X are blue and pink and similar in length, and they're all paired up along with each other, and the blue ones signify the chromosomes from dad, and the pink ones symbolize the chromosomes from mom. Together, they make up homologous pairs. The reason why they pair up is to conduct something called crossing over, and instead of defining it, I will show an example. So, to show that, here you can see that the alleles of both parents are as capital A, B, C, A, B, C, and then lowercase a, B, C, A, B, C. And in crossing over, what happens is that they join together and almost exchange hands. So we can see that this little c would go over here, and this big C would go over here. This crossing over helps to create new combination of genes and also ensures that genes are inherited from mom and dad and passed down generation by generation, and therefore plays a component in preserving genetic diversity as well. And other than that, however, prophase 1 is very similar to prophase in mitosis, as the nuclear membrane dissolves, the centrosomes move to the opposite poles of the cell, and spindle fibers are produced. And that's really just the fundamentals of prophase 1. And moving on to metaphase 1, there are two things to highlight here. Of course, the spindle fibers push all the homologous chromosomes and line them up in the middle. However, one thing to keep in mind is that they are lined up randomly. This process is called random assortment. And if you look at the picture, you can see that the homologous pairs are not lined up like all blue and all pink on each side. It is random, so we have some blue on the left and some pink there as well. This is what allows gametes to have different sets of homologous chromosomes and helps preserve genetic diversity as well. And another thing to keep in mind is that meiosis 1 expresses the separation of homologous chromosomes, while meiosis 2 expresses the separation of sister chromatids. And moving on to anaphase, this does not really need much of an explanation, but really all that happens is that the homologous chromosomes are pulled apart as the spindle fibers contract, pulling them to opposite centrosomes. Just a bit of a fun fact is that when an individual has something like trisomy 21, oftentimes it could be a result of the spindle fibers failing to pull apart the chromosomes. And next is telophase, and here the chromosomes have arrived at each centrosome, so nuclear membrane starts to be produced once more. Spindle fibers dissolve and the centrosome starts to kind of just float around. Although this picture does not express this other aspect very well, but the chromosomes do also decondense. However, since meiosis 2 happens right after, this step is quite short, so it wasn't really demonstrated in the picture. However, we can also see that the cell elongates in something called a cleavage furrow, which is kind of this pinch-in that happens around here. And basically this cleavage furrow forms, which then goes all the way through to separate the cell during cytokinesis. And here we can see cytokinesis, the cleavage furrow splits it. And the main thing to take out of this is that meiosis 1 separates homologous chromosomes, conducts random assortment, and crossing over. Meiosis 2 is almost identical to mitosis, and also at this point we can see that the two daughter cells have 16 chromosomes. And okay, now it's time for prophase 2. And same as prophase 1, the chromosomes condense, the nuclear membrane dissolves, centrosomes go to the opposite poles, and spindle fibers are formed. In prophase 2, crossing over does not occur as there is no other homologous pair to actually conduct it with, and also because it has obviously already happened. Also, as the cell moves from meiosis 1 to meiosis 2, it is important to note that the DNA does not replicate again. And metaphase 2 is just when the sister chromatids are lined up in the middle. At this point, it does not matter how the chromosomes are lined up, as no matter what, the chromosomes are paired as sister chromatids, so one will always go to one daughter cell and the other will always go to the other one. And anaphase 2, the sister chromatids are separated as spindle fibers retract, pulling the centromeres apart. A non-disjunction can sometimes result in the separation not occurring properly, and hence one of the daughter cells having more of a certain chromosome than the other, and this is regarded as a non-disjunction meiosis 2 generally. And telophase 2 is the same thing as in 1. The cell elongates, the nuclear membranes reform, spindle fibers disappear, cleavage furrow starts to dig in. But one thing to keep in mind now is that all of the 4 daughter cells have 8 chromosomes. We know the diploid number to N was 16, and we know that these are 8, which means they are N chromosomes. This therefore references that the daughter cells from meiosis 2 are haploids. And finally, this picture is kind of tilted, but after the cleavage furrow digs in, we get 4 gametes. The reasoning for why there are gametes is simple, as one gamete from dad, which is sperm, combines with one from mom, which is the egg, to form a diploid number of chromosomes. And the main thing to keep in mind with meiosis 2 is that it separates sister chromatids instead of homologous pairs, and that is all. And yes, thank you for watching.